So hello everyone, welcome to the Transnational EV Series, and thank you once again for joining us. My name is Débora Ferreira and I'm a postdoc at the University of Minho from Portugal. Today I have the pleasure of moderating this talk alongside my colleague, Carlos Jesus, that is a PhD student from University of Coimbra, also from Portugal. It is a great pleasure to introduce our special guest, Dr. Maria Morille. She is an associate professor in the Montpellier University and Institute Charles. Sorry, since 2012. She conducts multidisciplinary research working on the design of innovative biomolecule delivery systems with a particular focus on the coordination between the physicochemical properties of nanovectors and their impacts on biological performance. Her group, which brings together chemists, biologists, and pharmacists, is working on the development and fine characterization of lipid and/or polymer-based nanocarriers for the delivery of nucleic acids, as well as on the engineering of natural nanocarriers as extracellular vesicles. She will present today an overview of what her group have been doing on the surface modification of EVs to improve their administrability and make them more rational as therapeutics. I kindly ask to the audience to submit their questions in the chat box. These will be addressed at the end of the session. And Dr. Marie, thank you once again for accepting our invitation. We are excited about your talk and eager to hear your insights. You may now begin your presentation. Thank you.
Thank you. Thank you, Deborah. Thanks a lot for the invitation too. So I'm indeed Marie Morille. I'm associate professor in Montpellier University. And I will talk today about the link that exists between polymers and EVs, and especially the interest of polymer or PEG alternative for EV surface modification. So first, I will talk a bit about our wonderful Montpellier University. So for those who don't know Montpellier, it's in the south of France. It's 3 hours by train from Paris. And we are among the first university in Europe. Montpellier University was founded 8,000 years ago. And so, of course, we have this wonderful historic buildings, but we have also more modern buildings. Sorry if I can hear some attendee here. Oh, never mind. So Montpellier University is a really nice place to work. And aside that, if you join Montpellier as a postdoc or as a student, you could also enjoy the 300 sunny days we have per year and all the nice streets and all the nice environment we have. We are also close to the Mediterranean Sea. So yeah, don't hesitate if you want to join us to come for science, but also for the nice life we have here in Montpellier. So let's go in the research now. So I'm part of the Institut Charles Gerhardt of Montpellier. It's a chemistry institute, and here we work on development of different materials for energy, environment, and health. And so I'm part of the macromolecular chemistry and material department who is working on polymers. So it's the polymer department of ICGM. And among all these chemicists who work on these different application of polymers, we are a few who work on polymers for health application. So of course, the polymer design and formulation are the core of our activities. Again, there is chemicists, biologists, and pharmacists who work together for developing tissue engineering systems. So we worked mainly with hydrogels such as collagen, gelatin, chitosan, polyoxazoline. We will talk about this polymer later a lot. And we also work on shaping method of these polymers using 3D printing, stereolithography as an example, but also microencapsulation and so on. And in parallel to these tissue engineering activities, we also work on drug delivery system, especially nanomedicine, for biomolecule encapsulation, nucleic acids, but also proteins, as well as poorly water-soluble molecules. And we are also using polymers to modify the surface of lipid nanoparticles or extracellular vesicles. And of course, we have strong links between this nanomedicine part and the tissue engineering part, because we are using lipid nanoparticles to functionalize the biomaterial we can develop. So we are also working with what we call gene-activated matrices or protein-activated matrices to have a nice microenvironment for regenerative medicine as an example. And we especially work on cartilage regeneration, but I won't talk about that today. So the link between EVs and polymer is not maybe obvious for everybody, but you should know that there is, and since quite a lot of time, there is strong links between polymers and EVs at different steps of EV research. Of course, you have a lot of polymers to produce EV, looking at cell culture flasks or microcarriers you can use in bioreactors, even collecting tubes. You can also find polymers in EV isolation or detection for the chromatography, for the mobile phase of chromatography, stationary phase, or for the isolation method based on PEG polyethylene glycol precipitation. All the hollow fibers that you can find in separation devices are also constituted of, are also built with polymers. So there is really a lot of polymers in the EV environment. And of course, for therapeutic application, there is also different strategy. Surface modification, we will talk about that in a few minutes, but polymer can also be used to create hydrogel, to have a reservoir of EVs, or EVs can also be associated to fibers or microcapsules and so on. So if you want to know more about the link between polymers and EV, don't hesitate to look at this review we published 2 years ago in JEV. We will give you more details on this strong interaction there is between EVs and polymers. So going back to EVs, of course, you probably all know a lot about EVs. You know that these are vesicles that are present in all body fluids that are produced by all cells. These vesicles are the key player in intercellular communication, and for that they are naturally equipped to cross extracellular and intracellular barriers thanks to their composition of various lipids and proteins and so on. And they are also carrier of biomolecules able to trigger a biological response in target cells. So theoretically, it makes them the perfect drug delivery system. That's why we can think, and other people also think, to use them as drug delivery systems for the delivery of various molecules, biomolecules, small chemicals. But they can also be interesting for their interesting properties when they are produced by cells that have therapeutic interests, such as mesenchymal stromal cells, which are well known to be antifibrotic, immunomodulatory, and so on. So that's the good part of EVs, but in the real life, we also all know that EVs are a heterogeneous population. That's part of them. It's really, I think that their interest is also linked to this heterogeneity. It's not easy to produce EV, it's not easy to store EVs, it's also not so easy to load or to associate an exogenous molecule of therapeutic interest inside EVs. And one of the main limitations we will talk about today is their plasma instability if you want to administrate exogenous EV sources, because we will see that there is a really short half-life and a rapid relocalization in liver and spleen. So in our group, we have three main axes, research axes to try to answer to this limitation. So one of our main goal is to avoid genetic engineering of producing cells. As you saw, we come from a chemistry lab and a physicochemical. We have a physicochemical background working with synthetic nanoparticle mainly. So we aim at using only, for now, pharmaceutical physicochemical methods, and we worked on already purified small EVs to try to increase their therapeutic potential. So no genetic engineering of producing cells, and we also try to don't modify the proteins that are present at EV surface when we work on surface modification. So our three main axes are the pharmaceutical engineering. So what I call pharmaceutical engineering is really linked to the handling, to the practical manipulation of EVs. So how to store them, how to administrate them. So 5 years ago now, we published a paper that showed that EV, depending on the buffer in which you store your EV, you can really increase the recovery after freezing, as well as the recovery after freeze drying. And for now, we are currently working on EV formulation for improving their recovery after nebulization, especially. Of course, we are also working on different therapeutic molecule loading, proteins, nucleic acids mainly. I won't talk about that today. And we are developing surface modification of EVs for the control of their behavior after administration, but also to think about how we can associate them to biomaterials. So this presentation today will be really focused on the surface modification with polyoxazoline to try to increase EV half-life. Why? Because we know, as I said a few slides ago, we know that there is a really low stability of EV in the plasma. It's sometimes not easy to compare studies because there is huge variation in EV origin, amount of injected EVs, labeling methods, and so on. But even with all this variation, whatever the study, the main conclusion is that the circulation kinetics of exogenous EV are really low. You can see here that it's less than 0.01% of the injected dose that is still in the plasma after 1 or 2 hours after administration. And I could continue this list. It's a really well-established fact now. Even if sometimes in the literature we can see different facts. So these EV are really quickly eliminated from the blood, and where they go afterward, they will quickly recollise in the liver and spleen because they are handled by the macrophages mainly, so Kupffer cells in the liver and splenic macrophages in the spleen. So as we all know that, there is also some strategies that have been developed to try to increase EV stability in blood. So there is these 3 main strategies. Maybe there is other ones that I don't know, but I chosen here to highlight these ones. So one of the strategies is to use the CD47 don't eat me signal. So it's a natural process that is used by red blood cells or platelets that will interact with the SIRP alpha receptor on macrophages to and that will avoid their phagocytosis by macrophages. So several teams chosen to try to overexpress CD47 at the surface of EV using bioengineering method or using exogenous methods, so after purification of EVs. And this indeed increased the circulation time, even if this increase was quite limited, sorry. But there is also the side effects due to the fact that, as I said, this is a natural process. And so if you overexpress and if you inject a lot of EVs in the circulation, it will saturate the SIRP receptor, and it could lead to a decrease to an increase in red blood cell or platelet phagocytosis by macrophages. So it could be a really important side effect. Aside the CD47 dotted listener strategy, there is a strategy that was designed by Samir El Andalusi group that you heard, I think, during the last SNEV meeting. So in Samir El Andalusi group, they developed this albumin binding domain. So they use this domain to decorate EV, also using either engineering or bioengineering. So linking this domain to different part of the tetraspanines or also LAMB2, sorry, or using a lipid anchor associated to this albumin binding domain. And thanks to this domain, when the EV are administered in the plasma, the albumin will associate to the EVs and will allow to stabilize EVs and to hide EVs in regard to macrophages. So this strategy that is that is efficient to stabilize the EV. So there is an increase in stability in half-life. You can find EV till more than 4 hours after administration, and after 2 hours there is an increased accumulation in lymph node and tumors compared to non-modified EV. So it's really an interesting improvement in circulation time. But yet there is still less than 0.1% of injected EVs that were found after more than 4 hours after injection, and still a majority of EV relocalized in liver and spleen. So there is still improvements to do on this part. And the third method that is used is the coating with polymers. This strategy is often used for synthetic nanovector that are covered with well-known polymer that is polyethylene glycol. This polymer is hydrophilic, also really flexible, so it will create a steric barrier around the EVs. And this strategy was developed also was evaluated by several teams also in the EV field, in Peter Vader team as an example in 2016, and they evidenced that indeed it increased the stability with an improvement of EV plasma residence. EV were found till 60 minutes after injection, but yet, depending on the EV source, there is no tumor accumulation, and again, a rapid relocalization toward skin and liver. So all these strategies, as you saw, they gave interesting results, but yet we are still really limited in the stabilization of EV who are still quickly eliminated. So aside this fact, the polymer coating with EVs was exclusively done with polyethylene glycol. It's the same for the synthetic nanoparticles. All the nanomedicine from now are stabilized by this polyethylene glycol, was interesting both for the colloidal stabilization. It avoids aggregation, and also interesting to avoid adsorption from the plasma proteins. But we also know that healthy people also possess anti-PEG immunoglobulin, both IgG and IgM, and this is due to the fact that there is PEG everywhere in our daily product. There is PEG in cosmetics, in food, and also, of course, in nanoformulation or therapeutic proteins, because it's used to stabilize this nanomedicine or this protein. So what is the problem with this presence of antibody against PEG? It's that in the best case, there is a quick elimination of the PEGylated nanoformulation. So it's not a good point, but it's not too harsh for the health of the patient. So you have this accelerated blood clearance and it prevents multi-injection. So it's still a problem. But what is more an important limitation is that in rare case, you can have events of complement activation related pseudoallergy. So it's a syndrome that is abbreviated under the CARPA syndrome. And this could lead to a huge inflammation, and it could impact. It could have consequences on heart or lungs of patients. So this is a huge problem, especially because there is no millions of people who were vaccinated with PEGylated formulation. So we don't know now if this problem of PEG antibody could really be important in the next year. So that's why even if PEG is a really good polymer, it's really efficient to stabilize nanoformulation and proteins, but we also know that we need a PEG alternative because this polymer is really overused for now in the different products today. So it's in this context that in our lab, we chosen to modify EV surface, not with polyethylene glycol, but with an alternative to this polyethylene glycol that are polyoxazoline. So these polyoxazoline are also hydrophilic polymer, rather flexible, even if it's a bit less flexible than PEG. And to associate this polyoxazoline, we rely on the expertise of our colleague, so Vincent Lapinte, who is the expert of polyoxazoline in our lab, and of course Lauriane Simon. So all the work that I will present afterward will be is the work that Lauriane did during his postdoc, and she is now still in our institute, but working on other subject. But she really did a great job on this EVPOX story. So this polyoxazoline, as I said, are hydrophilic polymers. And so how to associate hydrophilic polymers to lipid nanovesicles that are EVs? We needed to find a lipid anchorage. So for this, we designed two lipopolyoxazoline, polymethyl oxazoline, during this project. So we have the CHEMS box who use cholesterol hemisuccinate encore, and we also use an hexadecan, a C16 POX, who use a linear carbon chain. So this C16 POX was developed during the PhD, during Lauriane PhD, under the supervision of Vincent. And so these two lipopolymers were associated to EV using co-incubation step that is called and it's a process that is called the post-insertion method. So it will rely on the affinity of the lipid part who will insert in the EV membrane. So it's a simple and really soft process. There is no harsh condition. It's a simple co-incubation. So of course, we optimize the POX EV ratio, the temperature, the time, the EV concentration, but it's a quite simple process. And so after this co-incubation, who was a bit different between the C16 and the CHEMS, the co-incubation was 2 hours for the C16 POX and 4 hours for the CHEMS POX. So after this co-incubation, we removed the non-associated POX using an ultrafiltration steps. And so with that, we have our poxylated EVs. So after that, the first step was to look at the physicochemical properties of these poxylated EVs. We shown that compared to the native EVs, we have a slight increase in size, but non-significant, looking at the hydrodynamic diameters, as you can see here. Of course, during each steps of this study, we compared the poxylation to the gold standard pegylation. For this, we use control, lipopeg control, that is a DSP PEG polymer. So you can see that there is no, whatever the polymer, there is no significant increase in size, but there is a significant decrease in zeta potential. So it makes sense because we have this coating around the EVs that change the surface charge of the EVs. And we also shown that either with POX or with PEG, there is no images here, but we have no structure modification. We keep the vesicular structure of the EVs. So after that, the next step, as we use mesenchymal derived mesenchymal stromal cells derived EV, we wanted to check that the poxylation didn't impact the immunomodulatory activity of these EVs. So for this, in collaboration with Farida Jouad in Montpellier, we looked at the effect of EV on activated primary murine primary macrophages. And what we saw 24 hours after treatment with EVs is that we have a tendency to decrease the pro-inflammatory associated markers. You can see here a decrease in CD80 in the CD80 marker, and we also have a decrease in TNF-alpha secretion. But we didn't evidence an increase in IL-10 anti-inflammatory cytokine. Aside the primary macrophages, we also had a look on leukocytes, and this was a collaboration with Kathleen Clark and Samuel Efferson in the laboratory of Ejun Wang in UC Davis. So in this context, we isolated PBMC, and when we selected the CD3 positive lymphocyte population, we saw that with the EVs and even more with the poxylated EVs, we had a decrease in CD3 positive cells. So it seems that the POX are interesting to increase the decrease in lymphocyte proliferation. So altogether, it's shown that at least the POxylation didn't disturb the immunomodulatory properties associated to MSCs, MSC-EVs. So next, we wanted to use these EVs as drug delivery vector in the context of cancer. So why we want to use this EV in this context? It's because we wanted to rely on two main facts. The first is what is called the enhanced permeability and retention effect. So it's a well-known process since that is really exploited for the synthetic nanoparticle. We know that in tumor there is a discontinuous endothelium. You can see here that there is holes in the endothelium due to the neoangiogenesis. And it's known since decades now in the nanomedicine field that if you manage to increase the circulation time of nanocarriers, you have more chance for them to accumulate in the tumor due to this leaky vasculature. And with that, you have a lack of lymphatic drainage who allow the nanovector to stay in the tumor environment. So this point is indeed working. This effect is really well established in mice model, but when nanomedicine went to clinic, it appears that it worked not so well in human because of the heterogeneity, depending on the patient, depending on the tumor. There is really an issue of reproducibility. And one of the main barriers is that we also know that in the tumor there is an important extracellular matrix deposition. This extracellular matrix is really stiff, and it's an important barrier for drug or nanocarriers penetration, even if we have this accumulation. But it's also well known that when you want to increase the half-life of nanomedicine, you are using the polyethylene glycol I already evocated. And this polyethylene glycol will avoid the interaction with macrophages, but it could also avoid the interaction with target cells and tumoral cells. So it's really a harsh to find a balance. Because you need to stabilize the vector to have a passive accumulation in tumor, but at the same time, the stabilization could avoid the interaction with your target cells. And that's in this context that the EVs can be really interesting, because we know that EVs have the ability to well diffuse in complex extracellular matrices. So it's a natural properties they have. So that's why we thought that in this context of tumor treatment, the EVs can be game changer and could increase the tumoral penetration, the penetration in the tumor microenvironment. So first, in a simple way, we just wanted to check if EV or poxylated EV were still able to interact with tumor cells in simple 2D condition. So for this, we stained EV with BID, the well-known lipophilic dye, who have drawback. We can talk about that later, but yet it's still allowed to look at to evaluate the interaction between EVs and cells. So after core incubation at different time points with the ID labeled EVs and 0.1 cells, we evidenced that there was no significant differences in EV internalization. Even with POX, we still have a nice internalization in cells, whereas it seems that it was surprisingly increased with polyethylene glycol. But so that was just a simple control, this co-incubation in 2 dimensions. But what was particularly interesting, even if we have to dig a bit on this part, it seems that after 3 hours of incubation, we have a decrease in colocalization between EV and lysosome compartments. that we used here, because we used here, I forgot that, but we used here marker of lysosomal compartment. And so that was really an interesting part. It seems that poxylation seems to maybe increase the endosomal escape. This is really preliminary result that we have to confirm, and we are looking at this. We want to look more seriously at this part. This was just interesting but non-expected result we had with this simple co-incubation. So at least the poxylation didn't avoid EV to interact with cells. And what was the most important part afterward? It was to check that they have the maintain the ability of EV to penetrate into extracellular matrices, especially the dense extracellular tumoral tumor extracellular matrix. Sorry. So for this, in collaboration with Jay Prakash in Twenty University, we looked at the capacity of EV and poxylated EV to penetrate in pancreatic cancer cells spheroids. You can see here in red the labeled EV, so still with DID, sorry. And so you can see that the native EV are able to penetrate in this spheroid. It's well distributed in the spheroids, which is not the case when we are using synthetic nanomedicine at all. They rather stay at the periphery of the spheroids. And so this interesting fact was not disturbed when we poxylate EVs. It seems that we have even an increased presence of EVs when they are poxylated, and they were well distributed all in these spheroids. So this poxylation seems to preserve the natural penetration properties of EV into extracellular matrices, and it seems that we have an increase of this penetration even with POX. So it was another good news. So afterwards, we wanted to look at the stability in vivo after intravenous administration. So in this context, there is a huge challenge that is the EV labeling for in vivo detection. So you may know or not, I don't know, but I wanted to explain a bit on this part, because there is different strategy that can be used for labeling EVs, relying on fluorescence detection, bioluminescence detection. You can also use inorganic nanoparticles such as superoxide paramagnetic nanoparticle to allow MRI detection. And there is also the radio labeling strategy. So all these labeling strategy have impact on EV, of course, because you can use either bioengineering, so working on genetic manipulation to associate bioluminescence signal or fluorescence protein on EV component, EV proteins. You can also use exogenous labeling methods, so using click chemistry as an example, or lipid anchorage of the labeling molecules on EV membrane. But whatever this strategy, again, it's really important to check that the labeling strategy is specific to EVs, because you can also really easily stain protein aggregates, and you also there is also a problem with the sensitivity of the labeling method, because currently in the EV biodistribution or pharmacokinetics studies, there is a huge amount of EV that have to be injected to be able to detect a signal. It's in the range of 1 to the 10 or 1 to 11 particles per mice. And so it's a problem because it's probably not really relevant in terms of the amount we can afford to use in therapeutic application, and it can even be non-really physiologically relevant. So for this reason, in collaboration, sorry, I forgot to talk about this point. So among these different methods, there is this really nice study that was carried on by Elisa Lazaro Ibanez 4 years ago, who compared different labeling methods. So if you don't know this work, I really invite you to have a look at it. And what is the main conclusion is that the radio labeling is one of the best methods to have a high sensitivity in vivo. And so that's why we wanted to use radio labeling in this work. And for this, we worked in strong collaboration with Julie Constanzo and Jean-Pierre Pouget in ICGM in Montpellier, who are specialists in radiotherapy. And so they designed EV labeling strategy using iodide to label EV. And by contrast to what was usually done, we didn't link the radio labeling to lysines, but we use labeling of histidine and tyrosine. After this labeling, I won't go in details on the labeling protocol, but after the labeling, we really discard the non-associated radio elements, and we checked the labeling efficiency, and the radiochemical purity was more than 98% for each condition. So after that, we use this method to look at the blood stability of EVs. And what we saw is that we, compared to native EV, we had a strong increase in blood half-life when EV were epoxylated, even if it was less important than the gold standard PEG. Aside that, we also checked at the hepatic and kidney impact. So there was no liver and no kidney impact after the injection of either native or epoxylated or pegylated EVs. So in both cases, we had an increase in EV half-life, and that was really interesting, even if with POX, to be honest, we were a bit disappointed because we saw that it was less interesting than PEG. But what was more interesting afterward, it was that when we looked at the tumor accumulation, 24 hours after administration, we saw that we had a strong tumor accumulation with epoxylated EV, especially with the ChemSpox encore. Whereas it was not the case at all for the pegylated nano formulation. Looking at the liver accumulation, we saw that with epoxylated EV, 24 hours again after administration, we have a decrease in liver accumulation, which was really significantly more important than native EVs. So altogether, we saw that this epoxylation allow an increase, even if the stabilization in blood was not as interesting as with pegylated EVs, we saw that the modification with polyoxazoline really allow to change the biodistribution, and with an increased tumor accumulation at 24 hours. So this was particularly interesting because using different polymers, we managed to have different biodistribution. And so there is now still a lot to explore because we can think to use different polyoxazolline length, because for now we just use quite a long polyoxazolline chain, but we know from the PEG field that depending on the length of the polymer and on the density of the polymer, you can have different organization. You can have with high density, you can have a brush which is particularly efficient to avoid the protein aggregation. Whereas if you have a longer chain, you can have a mushroom conformation, as you can see here. And this mushroom conformation is less efficient in stabilizing EVs. And that's the model in simple membranes, but we don't know much about what happens in complex membranes, such as the one we have in EVs, because we don't know if we have patches of polymers around the protein that are already present on EV surface, and so on. So there is really a lot to explore. And we want to investigate that now. I'm a bit late, so I will switch to the last part. In parallel to this surface modification with POX, we are also evaluating the protein loading, the protein association, using this lipid anchorage strategy. It was the work of Antonin Marquand, PhD, who just brilliantly defended his PhD a few months ago. And so using this strategy, we shown that we were able to associate a protein of interest to EV, that we were able to control the amount of protein we could associate to EV. This was with the goal of an intracellular delivery. So we evidenced that this strategy allow to have to efficiently deliver the protein of interest inside cells. And so, of course, this process, this strategy can also be used to control the association of targeting moiety we could add to the EV surface. In parallel with this EV work, we also work with synthetic nanoparticles, and we evidence that ChemSpox by their own can auto-assemble and form interesting nanovesicle who can have really fast intracellular delivery activity. And we are also working on on using this polyoxazoline for the stabilization of mRNA LNPs or siRNA LNPs. But I have no time to talk about that now. So I would like to thank all the people who work hard on this paper, on this study. Of course, Lauriane, as I said, but also all the lab members, as well as our collaborators, all the funding who contribute to this work, and of course you for your attention. And I would be pleased to answer to your question. Oops, sorry.
Thank you very much, Professor, for the very interesting talk and for sharing some data that are quite interesting for sure. We have a few questions. I might ask a question, but I'll leave it to the end. We can jump into the chat box and check a question here. So there is a question from Deborah in which she's asking for regarding the optimization of post-insertion protocol, which parameters have the greatest impact on the efficiency of the modification? Would it be the incubation time or temperature, anything else?
Sorry, there is I don't know if it's just me but I didn't hear you well. Deborah, you can
repeat the question.
Yeah.
Can you hear me?
Yeah, Melanie.
Yes, the question is, it's in chat box, and it's Deborah that is asking regarding the optimization of post-insertion protocols.
Yeah.
Which parameters have the greatest impact on the efficiency of EV modification? Will it be incubation time, temperature, anything else?
So it's in the paper, but the incubation time was 2 hours for C16 pox and 4 hours for CAMS pox. And the incubation temperature was 40 degrees, I think. We tried to avoid, we evaluated several temperature, but we saw that above 45 degrees, of course, EVs were not stable enough, and it could impact the EV integrity. But at 40 degrees, it was okay. So we really looked at also the number of lipopolymer we had per EVs, because in the first part of the study we use huge amount of lipopolymers, and we saw that so of course you can have micelles aside the EV, you can have the size of these vesicles, but it also really could disturb the EV stability. So we really work on this parameter, temperature, but I would say that temperature is really important to have enough fluidity of the membrane to allow the insertion, but the concentration is a really important parameter too.
And following this question, I might add just a little question. Did you notice any kind if these were kind of stable, this reaction, or did you lose a little of EVs after the reaction?
I'm sorry, but I think there is maybe I will stop sharing. And so you are talking about the stability of the EV.
Yes, in post-insertion modifications, if you manage to recover all the EVs, or you have any kind of loss of EVs, or maybe they will disrupt or something.
That's a really interesting question, because that's something we wanted to look at, looking at the amount of EVs that were really associated with POX. So it means that we needed a labeling method for the POX. And when we designed the lipo-POX coumarin, in this case, we use coumarin as a fluorophore to label POX. And it turned that when we were using this coumarin moiety, the CAMS POX were from auto-assembly, and we were not able to post-insert them in the EV. So when we wanted to tag the polymer, it led to a more hydrophobic part, and so it really changed the solubility probably, and the properties of the lipopox. So it was a huge problem, and it's something that we really want to address, because I'm the first one who want to have information about the quantification of the engineering, and so we really want to find a method. We have some ideas, but to be able to have this information without using fluorescent labeling, because it really disturbed the property of the lipopolymer. But yeah, it's something that is planned for the future.
Yeah, great, thank you. So Carrie Ann, we have a question from Linoy that is asking about what was the particle numbers, EVs, or proof concentration that you use in your in vitro assays?
What was the EV concentration we used, you mean?
Yes, that were incubated with the cells.
So for the PONC1 cells, we were around 250 EV per cells. And it was around 200 also for the co-incubation with primary macrophages.
Okay. And Lee is asking about, so the structure of the lipid bilayer of the EV are bound by components such as cholesterol, lipid interaction, or even the tetraspanning. how does the POX insert itself into the lipid biolayer? Any mechanistic studies done in this aspect?
It's with the lipid part that the polyoxazine insert in the EV membrane. So that's one of the main reason we have this association, but we can't exclude that this hydrophobic part could interact with some hydrophobic pockets in protein that are at the surface of EV. So it's also something it's part of our perspective, really try to establish the relation between the structure of the POX and the different lipid anchor we can add, and the conformation we have on EVs, but it's really not an easy task.
So it could be mostly you think it's direct insertion, but it could have any other mechanistics behind.
Yeah, we can't exclude that you have interaction between even the hydrophilic part of the polymer can interact with proteins. So
exactly. So next question will be that what can explain the higher accumulation of the CAN POX in the tumor in relation to the DSP PEG after 24 hours? If you have any explanation about this.
We know that the polyoxazine and the PEG have different behavior, even usually looking at cell interaction. Polyoxazine are really efficient to interact with lipid membranes. So it's something that we didn't observe with the PONC1 cells, surprisingly. But it's well known that the POX have specific interaction with cell membranes, and as we saw with the ChemSpox micelles, so aside DVs, but we really notice that there is this strong cell internalization. So there is something happening. We are also trying to really understand how it works and why it's so efficient, but there is something to do with the fact that it's well established that PEG have that pegylation disturb the ability of nanoparticle or EV to diffuse in extracellular matrices and to interact with target cells. It seems that it's less the case with POX. So probably it's not the same flexibility, it's not the same conformation at the surface. We don't know for now, but there is, again, still a lot to explore on this side.
I will add a question to this because I think it could be interesting, at least in my point of view. Do you think that depending on the proteins or the type of ECM that is there, that could have any big impact on this? I mean, if it could be like elastin or fibronectin or whatever.
It could, of course. It could really have an impact. And it's something that we can consider to look at. In parallel to the systemic, to the interest of POX to stabilize DV in blood, we are also evaluating it in the context of lung application. And so one of our goals is to look at the ability of epoxylated EV to diffuse especially in the lung mucus, and some preliminary results from that. It could be really interesting regarding also the literature, because some silica nanoparticles stabilized with POX shown that the diffusion in the mucus was really improved, and I think that with the EV POX it could be also interesting. So yeah, it's also among the things we want to look at different environment and try to figure out how the EV POX behave in these different environments.
Yes, a lot of possibilities.
Yeah, too much to do.
Exactly. Carry on. So Deborah was also asking, what kind of targeting moieties are you intending to explore in the future? if you have considered other strategies than proteins.
We have for now, so what we've done with Antonin PhD, it was another topic. It was really to deliver protein inside cells. But for now, a follow-up study linked to that is that we want to add a peptide. So it's in collaboration with colleague in Toulouse, Gaëtan Liga. We want to evaluate the targeting of CMV cytomegalovirus to target cytomegalovirus with the goal to decrease cytomegalovirus proliferation, multiplication. Sorry. So it's one of our goal of our perspective for now, and afterward I will we are also part of a big project that started in Montpellier, looking at the treatment of different autoimmune disease, the Immune for Cure project. And in this context, it could be really interesting to try to target EV toward different immune cells, to try to see if we can control the immunomodulation and so on. So it's another part of the project, try to control the interaction of EVs with a set of immune cells, macrophages, but also autoreactive B cells and so on.
So the next question is from Ref Kartani. And the question is about the isolation of EVs and the purification before adding the POX.
Indeed, I didn't give information about that part, but in this case, we were isolating EVs from cell culture media and using differential ultracentrifugation. It was the best compromise between purity and yield for us, and so we had this level of purity, even if again differential centrifugation is not perfect, but compared to just TFF isolated EVs, because we tried in the first place also to work with TFF isolated EVs, and it turns that we stabilized a lot of protein aggregates and it was not our goal. So yeah, it's really important to have a good purity, especially when you work with exogenous methods to engineer EV. You have to have a well-defined EV product at the beginning with not too much of protein aggregates.
Okay, thank you. So next question is from Enas. And the question is, will you ever use fluorescence labeling for in vitro characterization? Did you use the small EVs, exosome, or microvesicles? And how did you characterize the EVs in general?
So I think that's related to MISEF guidelines. I can't say that I'm using exosome or microvesicles because we don't have the methods to truly differentiate these two populations for now. What we are working with is the small EV or the 100K pellet population. And looking at the fluorescent labeling, yeah, we are also using fluorescent labeling of EVs, even if it's not perfect. We know that there is a problem of leaking of this fluorescent dye, but still, it's an easy labeling method for in vitro labeling of EVs. We are also working for different application with CFSE labeling, which is more specific to EVs and who have less drawbacks regarding the diffusion between lipid environments, especially working with on the application we have for the formation of hybrids vesicles between synthetic lipid particles and EVs. If you are using a dye that is diffusing easily between these two spaces, it didn't make sense. So in this context, CFSE is a good labeling method, but it really depends on what we do. But the thing is that you have to have in your head the drawback and the limitation of each labeling method. We know that there is no perfect labeling method, so there is no perfection in life anyway.
I think Enes is adding, continue to that answer, which markers you are using for fluorescent characterization. But I believe the method that you were speaking is general for lipids. It's not focusing on specific EV markers. Is that correct?
No. We were not using specific EV markers in this case. Yeah.
Exactly. Yeah. It's like a lipophilic
Dye.
staining.
Yeah.
Exactly.
Yeah.
Okay. Thank you. So I guess this is answer. So we have another question. We have 2-3 questions to go. We have a question from Anaïs. She's asking if you observe more aggregation on the EVs with the POX addition.
Hi, Anaïs. So we didn't observe more aggregation with POX, even with PEG. It was even a bit better in looking at all the recovery amount of EV we could add when we use freeze drying as an example, or compared to native EV, it was often that we have an increase in recovery and after the oxidation of pegylation. And it's well known because these polymers are also used to stabilize, to avoid aggregation of nanomedicine. So it's also worked well with EVs in this case.
And following to that question from Alexandra, how can we avoid having aggregates in this case?
Use POX.
Any recommendation?
Avoid having aggregates in a general way, you mean, in a general?
Yeah, I think so.
I would say that as on EV surface you have proteins, of course proteins will aggregate, so you can think to use surfactant. It's what is done in the field of therapeutic proteins, so you can have polysorbates or and it's already we are also using polysorbate for some stabilization buffers, and it was also published by other teams. But yeah, you can use some stabilizer, albumin or surfactant, with all the drawback that it also could have. But
and now Leticia asked regarding the isolation methods, if you think that the PEG-based EV isolation, would say maybe like ExoQuick or this kind of methods, if this could interfere with the POX PEG modifications.
Of course, because PEG isolation led to a lot of protein aggregates, and you can also have PEG that stays stuck on the EV surface. So I won't use these methods to especially to modify the surface of EV for 2 reasons. Because one, you have a lot of protein aggregates who can be confused with the EVs in terms of size, and you also have, as I said, this PEG that stay at the surface. We don't know exactly how much. So no, it's not something that I would use, but.
Yeah, I tend to agree with you because for this kind of modifications, I think it's quite important that the EVs are pure.
Yeah.
Otherwise,
even
we don't
if
know
we
what
can't
we
have
are
the purity,
modifying.
but.
And lastly, a question from Anaïs regarding the problems of the EV stability in the blood circulation. Do you think that the future of EV as drug delivery vector could be in fact more restricted to local applications and accessible tissue organs?
It's a great question, Anaïs. It's the systemic application or the intravenous administration. It's always the Graal, we could say, but regarding the nanomedicine field, it's been more than 50 years now that people are trying to design long circulating nanoparticles, and there is still no perfect solution. So probably for EVs, of course, the local administration can be a good option. That's why also we are working on nebulization for lung administration. But indeed, it could be something to consider. But yeah, EV is not always the best administration pathway, of course. But yet, it's still a big challenge. So that's why it's interesting to look at it.
Okay, thank you so much, Professor Marie. We have passed the time, so I think we can close the session. Thank you so much to everyone that asked a question. I would say if you have any other question, maybe it's better if you contact Professor Marie,
of course,
if Professor Marie doesn't mind. And thank you so much once again for accepting the invitation and for sharing your data. It was quite interesting. And I think a lot of questions show the interesting topic that it was presented here. Thank you so much once again.
Thank you. Thank you once again for the invitation. And indeed, don't hesitate to ask questions by email or to contact me if you need.
Thank you very much. Have a nice evening.
Have a good day or night or bye-bye.
Thank you.